Hello everyone, I am Jeremiah Salvador Tuliao from Bachelor of Secondary Education in Philippine College of Science and Technology. And I'm now here to give some lesson about genetics. So first things first, we must define what gene expression is. Gene expression is a tightly regulated process that allows cell to respond to its changing environment. Take note, changing environment. And within a cell, there's a thing we call DNA. And in DNA, there's a process inside of it in making proteins. That's our topic for today. We will talk about two key steps involved in making protein. These are the transcription and translation. So hope you guys learn and enjoy. In the first slide, we will talk about the first step of gene expression, which is the process of transcription. Transcription. It is a process that makes mRNA from DNA. It is also called the first step of gene expression. In a DNA transcription, it is a process that involves transcribing genetics information from DNA to RNA. DNA housed within the nucleus of our cells. It controls cellular activity by coding for the production of proteins. The information in the transcription in DNA is not directly converted into proteins but must first be copied into RNA. Always remember that, guys. This ensures that the information contained within the DNA does not become tainted. Okay? In DNA transcription, DNA in the initiation of DNA, the enzyme RNA polymerase binds to DNA at the promoter region. Here, guys. First, DNA unzips into two separate strands. A is the DNA helicase is the enzyme breaks H bond. And the number 2, free-floating RNA nitrogen bases in the nucleus pair up with unzipped DNA nitrogen bases. These are cytosine, or we call it C, pairs with guanine, or we call it G. Okay. Letter B, the uracil, or we call it U, pairs with adenine, or we call it A. And letter C, thymine, we call it C. We call it T. Pairs with adenine, we call it A. Always remember that T is only with DNA. Into our second slide, transcription of DNA. That's it, guys. After all pairing is done, that's it. We will see the transcription making process in protein. A single strand of RNA has been produced. Genetic code from DNA is transferred from our mRNA. We will see in the video, okay, the transfer to of DNA to mRNA. That's it, guys. And number 5, the code obtained from DNA lets the mRNA now which amino acids to pick up. Okay, that's it. The code is a set of 3 nitrogen bases, or what we call codon. transcription. Okay, that's the image of transcription. And we will go further about translation. What is translation? Translation, it is the information in a messenger, RNA, and uses it to build a protein. Actually, into to this translation, to be a little more technical. Why? An, an mRNA doesn't always encode, provide instruction for. Instead, what we can confidently say is that it always encodes a polypeptide or chain of amino acids. Always remember that translation, it is a conversion of RNA into amino acids sequence that makes protein. Later on, we will talk about the difference of transcription and translation. Just wait for it. And the mRNA leaves the nucleus and enters the cytoplasm. The ribosomes attach to mRNA, and the tRNA, carrying anticodon, picks up the correct amino acids and carries them to the mRNA strand, forming the protein. Like the example, the tRNA carries GAU anticodon and looks for CUA on mRNA. You will see to the picture. Okay. The next slide. Okay. That's the sample of translation. There's some definition about translation. In this picture, we will see that translation is the direction which continues going in the right side. And the key differences of transcription and translation. Here we are. So we will see the basis of comparison the transcription and translation. So first we will see the definition of transcription. Transcription is the process where the genetic information on a DNA strand is transferred into an RNA strand by a series of polymerization reactions catalyzed by enzymes called DNA-dependent RNA polymerases. And in the other hand, translation. The translation is the process of protein synthesis where the information on RNA is expressed in the form of the polypeptide chains. Into its gene expression, the transcription is the first step in gene expression. The translation is the second and final step of gene expression. Always remember that. And we will go to occur. Transcription occurs before translation. Translation occurs after transcription. Okay? And let us talk about the precursor. The precursor of transcription is the non-coding and antisense DNA strand. And in the translation, the precursor of translation is the mRNA produced from transcription. Into its raw material, the raw material of transcription is the 4-base pair of RNA, which are the adenine, guanine, uracil, and cytosine. Into translation, the 20 amino acids are the raw materials of translation. Always remember that, guys. We go into initiation, the recognition of a specific DNA sequence, term promoter sequence initiation initiates transcription that signifies the beginning of the gene. Well, in the other hand, the binding of mRNA initiates the translation to ribosomes. Next, elongation. The elongation of RNA sequences occurs by the binding of complementary base pairs to the new sequence, while the translation is the elongation of protein occurs by the binding of amino acids. Okay? The product of transcription is the mRNA molecule which is complementary to the DNA strand, while the product of translation is the peptide sequences encoded from the mRNA sequence. The synthesis, the synthesis of transcription results in the synthesis of RNA sequences. Translation results in the sequences of proteins. In the site of each changes, transcription occurs in the nucleus of eukaryotes and in the cytoplasm of prokaryotes, where the enzymes and regulation factors are present. While, the other hand, the major enzyme that is responsible from translation is acetyl or tRNA synthetase. The regulation of both, the transcription is regulated by various transcriptional factors in eukaryotes and by operants in prokaryotes. And on the other hand, the translation control is mainly brought out by the binding of ribosomal units to the translation complex. And the modification or the post-events modification of each changes. Post-transcriptional modifications include the editing of pre-mRNA, the result of transcription by the process of splicing before the mature mRNA reaches ribosomes from translation. And the post-translational modifications involves the folding of polypeptide chains to the three-dimensional configuration. In the last slide, the detection of both the process in transcription can be detected by the methods like RT-PCR, DNA microarray, in situ hybridization, and northern blotting. In translation, similarly, the process of translation can be detected by methods like Western blotting, immunoblotting, enzyme assay, protein sequencing, etc. And in inhibition, it is inhibited, the transcription, it is inhibited by some antibiotics like rifampicin and 8-hydroxycholine. And in translation, it is inhibited by antibiotics like tetracycline, chloramphenicol, streptomycin, erythromycin, anisomycin, cyclosamide, or etc. So these are the key differences of transcription and translation. For further informations about the what we call the key differences of transcription and translation, you will just update or beep me on my Facebook, Jeremiah Salvador Tuliao, And also, okay, I will just open up my camera. Ding! So I hope you learn some of this topic about genetics, which are the key differences about translation and transcription. It's hard, but we survive. Yes. And thank you for listening. And I hope you will watch again to my fourth video, which talks about the mutation or the gene regulation. Thank you and God bless.